stability of messenger RNA and codon choice. If all the requirements for protein synthesis or translation are available, in other words, we can say that if translational machinery is there, even then the rate of expression of the cloned gene, it is dependent upon the stability of the messenger RNA. So there must be a steady state level of messenger RNA that can be deflected in terms that there must be a balance between the messenger RNA that is being synthesized and degraded. If we have strong promoters that can maximize the expression of the cloned DNA, it is equally good. But on the other hand, if we manage the time of degradation of messenger RNA or to enhance the stability of messenger RNA, it is equally good. So in case of messenger RNA degradation in the biological system or in the living cell, combination of two enzymes, they determine the stability of messenger RNA. One of the group of enzyme is the endonuclease and other is 3' exonuclease activity. So both these two enzymes, they can attack the messenger RNA and degrade it. On the other hand, if messenger RNA it is stable in vivo, के biological system में अगर किसी messenger RNA की जो stability है वो ज़्यादा है, so even then it is susceptible to degradation by these two enzymes. But its susceptibility, it will depends upon the sequence of the nucleotides in the messenger RNA, its secondary structure, it is also very important, and the association of messenger RNA with macromolecules like the ribosome, it can also influence the stability of messenger RNA in the biological system. In the biological system or in vivo, the degradation of messenger RNA, it is mainly mediated by two type of 3' to 5' exonuclease activity or enzymes like the RNase 2 and polynucleotide phosphorylase. So these two enzymes, they are important in the degradation of messenger RNA in the biological or in the living system. These two enzymes, they can act on the messenger RNA and degrade it. But if messenger RNA, they have secondary structure at the 3' end, then the degradation, it may be impeded or it may be slowed down. So because of this reason, ये ज़रूरी है कि पहले जो एंडोन्यूक्लियस एंजाइम की कैटेगरी है वो पहले मैसेंजर आरएनए को डिग्रेड करे ताकि 3' एंड एक्सपोज हों और देन 3' एक्सोन्यूक्लियस एक्टिविटी में बी देयर. लेकिन कुछ ज़्यादातर मैसेंजर आरएनए जो हैं वो 3' सेकेंडरी स्ट्रक्चर की वजह से रेजिस्टेंट होते हैं. तो इस पर्पज़ के लिए एंडोन्यूक्लियस की जो एक्टिविटी है ये बड़ी ज़रूरी हो जाती है. Now, there may be different type of endonucleases that can degrade messenger RNA, and one of the category is RNase E. So this RNase E, it can increase the turnover rates or the degradation of the messenger RNA. On the other hand, if we use the mutants that are defective in RNase E biosynthesis, then the messenger RNA degradation, it may be slowed down. But it has been observed that even the mutants of RNase E, they are able to degrade messenger RNA. The reason is that the living system or the microbes, they are not only containing RNase E, but in addition to that, they contain other types of RNAs, like the RNAs K, RNAs M, and RNAs M and R. So these are different type of RNAs that can degrade messenger RNA even in those mutants that are deficient in RNase E. So the combination of endonucleases and 3' exonuclease activity, they are very important to determine the stability of messenger RNA in vivo. In addition to the stability of messenger RNA, other factor like the codon choice during the process of protein synthesis, it is also very important. We have the idea that the degeneracy is there in case of triplet codon for different amino acids. या हम ये कह सकते हैं कि एक अमीनो एसिड के लिए एक से ज़्यादा कोडॉन होते हैं, and this situation we can call it as the degeneracy of the genetic code. अगर degeneracy है तो कौन सा कोड जो है वो यूज़ होगा, it is also very important in order to determine the rate of expression of the cloned gene. And even in the natural genes, the selection of the codon in case of degeneracy, it is non-random. It is not random, rather it is non-random. That is, selectivity process is there for the codon choice in case of degeneracy. And this codon choice, again, it depends upon two factors. तो पहला फैक्टर ये है कि ट्रांसफ़र आरएनएस की अवेलेबिलिटी की रेशियो क्या है सेल के अंदर, ये एक इम्पोर्टेंट फैक्टर है. और जो सेकंड फैक्टर है कोडॉन चॉइस में वो ये है कि जो थर्ड बेस है वो पिरिमिडीन है कि नहीं है, कि वेदर इट कंटेन साइटोसिन और यूरासिल. सो दिस फैक्टर of the presence of pyrimidine at third position of the codon, it can also influence the expression of the cloned gene. So this is not only related to degeneracy of the genetic code and its selectivity for gene expression, rather the stop codon, they can also determine that whether gene it is going to express highly or not. For example, if the stop codon is UAA, then it was observed that the gene expression it is relatively high as compared to other stop codon like UAG or UGA. So even the stop codon, it can also determine the expression of cloned gene.